Hello, my name is Detlev Boison. I'm professor and vice chair of research and training for the Department of Neurosurgery at Robert Wood Johnson Medical School. I'm also a member of the Leadership Council of the Brain Health Institute. The focus of my research is epilepsy, which affects 80 million persons worldwide. Despite the constant development of new anti-seizure medications, 1/3 of all persons with epilepsy remains refractory to treatment. Current anti-seizure medications are designed to provide symptom control, but are not able to treat epilepsy and its comorbidities comprehensively and have never been designed to treat the cause of epilepsy. So therefore, novel approaches for the treatment and prevention of epilepsy are needed. My research on epilepsy is inspired by the evolution of life on our early planet Earth, as I have outlined in a recent article in Nature Reviews Neurology. Life started with biochemistry, metabolism, and with RNA. One of the founding molecules of life in the primordial soup of early planet Earth was adenine, which resulted from the fusion of 5 molecules of hydrogen cyanide. Adding a ribose yielded adenosine, and adding the phosphates yielded AMP, ADP, and ATP as part of the founding molecules of life. 1/4 of all RNA is composed of adenosine, and messenger RNAs have poly-A tails. If you want to construct a living system out of scratch, the most important consideration is energy homeostasis. Whenever there is an energy crisis, energy needs to be conserved. The simplest way to design a regulatory system is the following. Whenever there is an excessive consumption of energy, there's a drop in ATP, and the resulting formation of adenosine is used to suppress energy-consuming processes. In that regard, an epileptic seizure is a state of excessive energy consumption. ATP is degraded to adenosine, which is then used to stop the seizure to conserve energy. Therefore, adenosine is the brain's endogenous anticonvulsant and seizure terminator, and we have shown that therapeutic adenosine augmentation is a powerful approach for seizure suppression. We also made the discovery that adenosine regulates DNA methylation. This again makes sense from an evolutionary perspective. If you want to invent gene regulation, you cannot start with transcription factors which need protein kinase pathways and G-protein coupled receptors which all need their own genes to be regulated. The simplest solution to gene regulation is adding and removing methyl groups, and we discovered that this process is under the control of adenosine, which is regulated by adenosine kinase, and specifically an isoform of the enzyme expressed in the cell nucleus. We found that insults to the brain which trigger epilepsy, such as traumatic brain injury, cause a maladaptive overexpression of adenosine kinase. We have also shown that overexpression of adenosine kinase drives the epileptogenic process that turns a healthy brain into an epileptic brain. Part of the underlying mechanism is that increased adenosine kinase in the cell nucleus drives maladaptive DNA methylation, which plays a role in the epileptogenic process. Based on those principles, we launched a drug discovery program to develop novel small molecule adenosine kinase inhibitors which restore metabolic and epigenomic homeostasis and thereby prevent the development and progression of epilepsy. Our data show that therapeutic adenosine augmentation can prevent the development of epilepsy in several of our model systems. We are very excited that we might be in the position to develop a therapy that is able to prevent epilepsy and its progression. All of this work would not be possible without having a fantastic research team of collaborators. And of course, this research needs substantial funding, and we've been lucky to be supported by the NIH and the Department of Defense and the Cure Epilepsy Foundation. Thank you for your attention.